Hello, class. I'm Professor Angulo, and today we're going to cover slides number 38 to 60 in packet 1.2. So sorry that somebody's trying to get in now. Okay, so signaling by hormones. Hormones don't get into the cell, and therefore there has to be a signaling mechanism. And And there are various mechanisms for hormones. We're going to see here only a few. So we're going to continue because there may be different signaling mechanisms for the same hormone in different tissues. So, for example, one very common, for example, all the neuropeptides and many of the other kinds of hormones, they use the G protein mechanism. The G protein mechanism, excuse me, uses this seven transmembrane alpha helical domain that anchors the receptor to the membrane. Binding occurs on the extracellular side, the amino terminus of the receptor, and then the G protein is bound to the C terminal domain, which is on the inside, the cytoplasmic side. But then there are others that we're going to see in more detail here, such as the growth hormone mechanism, which is very unique, and also the AMP, the atrial natriuretic peptide. So this is just a sampling. You can follow the domains here. AMP it's just one gene, one polypeptide, but it has a kinase domain and the C terminals, and it also has a guanylate cyclase to make cyclic GMP to make the second messenger. So the beta adrenergic receptor, which was the first that became well characterized, is coupled to excuse me, a G protein called GS, alpha S. S stands for stimulatory, and I, for example, stands for inhibitory. I think you know already about G proteins from biochemistry, and just a little refresher. The alpha subunit is what is unique. The beta gamma is the same. For many years, it was thought that beta gamma didn't do anything. It was just part of this mechanism that turns on the receptor and then turns it off. However, now we know more and more examples of beta gamma signaling, especially in the nervous system. Uh, so, so we're not going to go into details on beta gamma signaling. So for us, it's got to be mainly alpha. The important thing is that alpha activates an effector, in this example, adenylyl cyclase, which is then going to produce cyclic AMP. Alpha I stimulates or inhibits. It stimulates the inhibitory mechanism. So what it does is it inhibits cyclic AMP formation. So now here you see one pushes, the other is pulling back. And they're examples by different hormones, which affect an inhibitory mechanism. Another one stimulates, and they're on the same cell. So it's a way of maintaining some kind of homeostasis. So let us move on and see how it begins. So the receptor binds the first messenger. First messenger is the hormone. That causes a change in conformation. And what happens is that then the G protein gives up guanine triphosphate, it picks it up, gives up the diphosphate, which that energy was used to reassociate the complex. But in the initiation of the process, it exchanges GTP for GTP, and now the two dissociate. So alpha then activates an effector. In this example, adenylyl cyclase, production of cyclic AMP. Uh, then you can see that the GTP is still bound. You're going to use the energy in the GTP to be able to reassociate the complex. It's an energy requiring process. So the GTPase function in the G protein will reassociate the complex, and then the hormone, if there's more hormone, it'll bind it, and you go through another round. Now, let me make something clear, because some of you may have more biochemistry, and that is this mechanism we accept it here as the standard mechanism. This is not signaling. This is not a biochemistry class with the emphasis on signaling by G proteins. But there are, for example, adenylated cyclase varieties that are not bound to the membrane. They are cytoplasmic, and those also get activated. So there are other mechanisms operating. So by ignoring them, we're not, or by not giving them prominence in the course, doesn't mean that they're not important. They are very important, but we need to have a standard mechanism because, as I said, this is not G protein signaling biochemistry. So now you kind of understand this kind of limitation that we have imposed on ourselves. So now, important phenomenon about hormones, and that is hormones not only activate to produce a physiological response, there is amplification as you go along. For example, if you look at the example of binding of epinephrine to its receptor, the beta adrenergic receptor, you activate the G protein, second messenger is produced, and you go down activating kinases and other components. So one hormone molecule, in this example, produces 10 to the 7th glucose 1 phosphate molecules. This is a huge amplification factor, right? It takes no thinking to notice this. And the question that I can ask you is, and why is this so? How can the hormone do that? The hormone simply activates a cascade composed of enzymatic steps. All of these are enzymatic steps. What happens with enzymes? They are not consumed by the reaction, they catalyze. They turn over hundreds of times in 1 second. So that's the amplification effect, which can be huge. So this is how nature has set up this pathway, something that we couldn't design anything to work like that. So beta adrenergic does this, as we will see later. Glucagon in the liver does the same thing, binds to activates the same cascade. That's another important concept, and that is these cascades, intracellular cascades, are modular. So here, the beta adrenergic receptor, you could say, well, this is happening in some cell in the body. Glucagon in the liver will activate the exact same cascade. All you do is you use a different receptor, right? Which then couples to the same type of G protein. So there's modularity involved here also. So now we take a look at something like, how about the alpha 2 adrenergic? This is still epinephrine. It is still the same hormone. But now it is doing something with GI. See, the G proteins are also modular. So, and how does that happen? Again, I have to kind of qualify the answer because there are more than one mechanism by which this can happen. Mechanism we choose is equilibrium. So when you activate a G protein that doesn't couple to adenylyl cyclase, for example, the enzyme that produces the second messenger, what happens is that now you have excess amounts of beta gamma inside the cell, right? Because you did not activate the same enzyme, adenylyl cyclase. So eventually what happens is that beta gamma has a greater chance. That's what we call the equilibrium phenomenon of hitting alpha S and taking it away from, from the effector, adenylyl cyclase, thus reassociating the protein and turning it off. So this is how GI inhibits cyclic AMP formation. It doesn't do it directly by binding to adenylyl cyclase and inhibiting adenylyl cyclase. Adenylyl cyclase is not active unless it binds alpha S. So it simply takes away alpha S. That's the equilibrium explanation. There are other mechanisms involved, but again, can't go over all of them. Now, we're still with epinephrine, the alpha 1 adrenergic receptor. This one is GQ, and GQ is coupled or activates phospholipase C. Here we use as an example the membrane kind, but there are also phospholipase Cs that are soluble. They're variants. And the interesting thing here, or what is different here than the other two that we just met, is that you produce two second messengers, diacylglycerol and inositol triphosphate. Diacylglycerol activates protein kinase C, and then protein kinase C has effects which are fast reactions and also slow reactions, meaning activation of transcription. Now, inositol triphosphate binds to its receptor on the endoplasmic reticulum. The endoplasmic reticulum is the intracellular storage site for calcium. So the IP3 receptor is a channel. And then the channel, when it binds IP3, opens, as you have seen in the examples at the very beginning of part one. And calcium comes out. Calcium then activates calcium, calmodulin, protein kinase. There's different variants. Again, there's like 5, 6 different variants. And even those then can have subtypes. But the important thing is this is an enzyme which is going to phosphorylate factors that bring about the fast reaction, like opening a channel, etc. But also those slow reactions because it can activate a transcription factor that will then cause genes to be expressed. So you're beginning to see how these responses really involve activation of systems and how these systems of G proteins are modular. They can be found on the same type of cell. So now we're going to look at a kind of signaling that is very unique. And that involves the growth hormone receptor. The receptor is a monomer. It's one protein product, one gene product. And when it binds growth hormone, which comes from where? From the anterior pituitary, right? The receptor then dimerizes. The dimer then undergoes a change in conformation that the C domain, the intracellular domain, attracts a kinase, the Janus kinase, type 2. So the kinase binds, but the kinase then autophosphorylates when it is bound to the growth hormone receptor. And then when it autophosphorylates, it recognizes domains on the C terminus and phosphorylates those. In that state is when the receptor signaling cascade or transduction cascade attracts STATs, which are transcription factors. Then the kinase will phosphorylate STAT, and now STAT is able to recognize its promoter site and causes the transcription of genes. What are those genes going to produce? This is kind of critical thinking, but you can guess. Well, you can not really guess. You can deduce it by reasoning. Well, what do you need for a growth hormone response? You know that already. Insulin-like growth factor 1. So obviously that's going to be one of them, but then more. We'll see all the others later in the course. In part 2, we spend a whole packet on growth hormone. So everything I said there is summarized for you right here. And insulin-like growth factor 1 is then produced There's more than one type of ILGF in the cell. You'll see what the second factor does later. And other factors that are produced in cells that respond, that respond to growth hormone, primarily in liver. So we'll see the detailed cascade and where things happen in part two better. But this kind of signaling, which is very unique, but it's very effective. Now we come to another kind of signaling, the classical steroid hormone or thyroid hormone receptor. The steroids can get into the cell because they're hydrophobic. So they cross the biological membrane, as you can see here, without needing any energy to do it. Then they can get to the nucleus as well. They can cross the nuclear membrane, and here they bind to an steroid hormone receptor. There are different hormone receptors, as you will see here soon, and then genes are expressed, sometimes de novo. And this was the classical mechanism. Then recently has been found that there are also the steroid hormone and thyroid hormone receptors on the membrane, and that they signal like a G protein mechanism. We still don't know what that is doing quite well, but it's fairly recent, so it takes time to characterize this. So now let's take a closer look at what's happening here. First, the different kinds of receptors and the structure of the receptor. The steroid hormone receptor is a family of receptors which arose in evolutionary time by gene duplication of that original gene. There is one for each hormone. For example, here H stands for human, the human variant, for glucocorticoid receptor, which is the involved in, among other things, in the stress response. Then the mineralocorticoid, aldosterone, the the androgen receptor, which is testosterone, the progesterone receptor, the estrogen receptor, the retinoic acid receptor, the thyroid hormone receptor. This also arose by the same gene duplication mechanism. There's also the vitamin D receptor. And then there's even one that we still don't know what is the ligand. Maybe they're like an appendix. Uh, we don't know that yet. We just haven't found the ligand for this COOP receptor. Now let's look at the domains. It has a DNA binding domain. The DNA binding domain is something recognized by each receptor. In other words, there's no cross reactivity. These receptors are very selective, and when they bind their hormone, they recognize only this DNA domain, which is where? It's in the promoter for the genes that it is going to activate. And it has a structure that recognizes a DNA binding element. We'll see a little bit more about that. Then it has the hormone binding domains. It has two of them. And the hormone is, again, the binding domain recognizes only its hormone. It doesn't cross-react with any of the others. So these are the important concepts here. So how does it really work? Well, the receptor has a DNA binding domain. If you don't cover it, it's going to be binding to the promoter, but doing nothing. And that can even interfere with mechanism of expression of the genes. So it is masked or covered by the heat shock proteins. This was discovered in bacteria when temperatures raised. In the incubator in bacteria, they produce this to kind of be able to deal with the stress of the elevated temperature, and that works for them. Now, that doesn't work for us, but nature uses the same thing in different places. It doesn't have to reinvent the wheel every time. So these heat shock proteins cover turn the receptor into an inactive receptor. Then the hormone, when it's there, it binds and it begins to dissociate the heat shock proteins. The receptors now are able to dimerize each subunit binding one hormone. And this is the mechanism by which it is now able to bind to its response element and the promoter. And you can see it here shown better. So the dimer with the hormone bound recognizes its response element, which is unique for each hormone. The important thing is that if you look at this, these two subunits are identical. So the response elements for the steroid hormones are palindromic. And again, you should know that from biochem, right? You can see it here. You can read this here from right to left, and this is the sequence. If you read the other strand of the DNA, now backwards, what do you have? The same. That's because these dimers are identical. So the response element is really the key to the gene that is going to be transcribed. It is acting really like an enhancer of transcription, but one that can be turned on and off by the hormones. Here's RNA polymerase with all of its accessory factors. So this case for the steroid hormones involves coactivation, and the dimer is a homodimer, identical subunits, and the response element is palindromic. Now let's take a look at something else. Here is the thyroid hormone. The thyroid hormone receptor is a heterodimer. One subunit is the retinoic acid receptor subunit. The other one is the thyroid hormone receptor subunit. There's also a repressor associated with the thyroid hormone receptor. What is T3, the thyroid hormone? Remember, T4 is a precursor to make T3 in tissues. Well, T3 then causes the displacement of the core repressor. And then the receptor, the heterodimer, can bind transcription factors and attract RNA polymerase II, and then begin transcription from the data box. And you see, this is very different already from the steroid hormone. Another difference is that the response element is not palindromic. Do it yourself. Read it left to right, and then go to the other strand and go right to left. And you'll see they're different. That's because these two are different subunits. One is the retinoic acid receptor, the other one is the T3. They associate with T3 bound to the complex. So now you see that you can contrast now yourself, but it doesn't end there, because you also know from biochem that heterochromatin is so compact that RNA polymerase II, which is a huge complex, cannot get in there and begin transcription. So heterochromatin is really silent, transcriptionally silent. If you bring about decondensation, then the polymerase can get in, and then it transcribes. It goes around these histone complexes which make up the chromatin. And what happens now with thyroid hormone? Well, thyroid hormone has an histone acetyltransferase that restores the acetyl groups and the histones. So when the acetyl groups are removed, it becomes condensed, not transcriptionally active. When you restore the acetyl groups, you change the charge. So now there is repulsion of charges. And that's how you cause the decondensation of the chromatin, and thus turn it into a transcriptionally active component. You see, thyroid hormone can do this. The steroid hormones don't do this. So one more difference between these two classes. Now we're going to look at things like, how about some heterochromatin, which is developmentally set. Well, that's why you can look at every cell in the body, and you know already that every cell in the body, if we look at the example of the gene for ovalbumin, every cell has the gene for ovalbumin. But cells in the ovary express ovalbumin, but cells in the liver do not. So why is that? Well, it could be because they don't have the estrogen receptor, right? This is under the control of estrogen. Okay, but that's not the fact. The fact is, liver cells have the estrogen receptor. So that means they should be able to make ovalbumin, but they don't. The reason is that the gene for ovalbumin is said to be heterochromatic in liver cells, in other cells of the body. But then in the ovary, it is euchromatic, it is open. And then the estrogen receptor binds estrogen, activates transcription, and thus you express it in ovary all the time. So this kind of developmental mechanisms, they stay for life. They usually do, except cancer cells do very funny thing. They activate genes that have been supposed to be silent for life. And that, cancer cells are like that. So if you have had cancer biology, then you know the mechanisms involved in this kind of events. So there's the explanation. So now we're going to talk about assays to be able to assess what is happening. What is one thing that you know already and you can deduce it with critical thinking, even though you will not know the mechanisms? My role here is to explain them to you. First, I need to tell you how. You have the assays that are available, the tools that are available. But let's look at the problem that has occurred. First, hormone dysfunction can be happening at multiple levels because you know already that it begins in the hypothalamus with the releasing hormones. Those are the ones that activate the anterior pituitary. Then it releases its hormone, and then that goes to the target tissue, right? So if you have a problem in the hypothalamus and you don't make the releasing hormone, you're going to have a deficit. What else can give you a deficit or a dysfunction, a pathology? Well, by the same token, you may get the releasing hormone from the hypothalamus. There's something going on with the pituitary, and it doesn't give you the hormone. Again, the target tissue is going to be responding like there's no hormone, and that's going to cause a deficit. So you see there are multiple sites. So what do you need to be able to really get a handle on this and be able to then diagnose a pathological state with something that is concrete, like hormone levels? Where's the problem in this hypothalamic pituitary tissue, target tissue kind of axis or cascade? The beginning, it was impossible to measure hormone levels, especially in plasma, but even in experimental settings. You're seeing already that hormones can function at 10 to the minus 11 molar concentration. That is a trace amount. You couldn't do it with any method available, say in the 50s, because they were colorimetric and you couldn't generate enough color to detect it in a spectrophotometer. So what you needed was an assay. Now, antibodies can be made or they can be isolated, and antibodies are present in very small levels, but antibodies are very selective. So you can make an antibody to the hormone, and the antibody will bind the hormone in 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 say in vitro, but you don't know, you have no way of detecting. So people knew that this could lead to something, but what? Well, it had to wait for radiochemistry had to get to the point and develop to the point where it could put a radioactive group or element on a substance, an organic substance. Bingo! Because now you have your antibody. It binds very low levels of of, in this case, hormone or antigen. But if you make it radioactive, you know the specific activity, right? Because radiochemistry developed the means to do this, the analytical means to do this. And so you now knew the concentration of the radioactive element, that's the hormone. You can then add samples from plasma, and if you have a lot of the hormone there, he would displace it. And then you can isolate that and you can count it. So you see, now you have a radio immunoassay. It had to wait for radiochemistry to be ready to provide the tools to do it. So what then involves? What is the radio immunoassay? That's what we're going to look at now. So radio immunoassay makes use of what you already know, because I introduced that already, uh, K dissociation, K association. So if you mix a radioactive antigen with an antibody, they will associate. And because the antibody is such a large molecule, you can trap it on a filter and then you can count it, right? So the more radioactive antigen you use, which now you can replace that in your mind as hormone. There you have it. Now you have your complex. But remember that this is subject to also dissociation. So this will come off, and then the antibody will simply bind another antigen. If all you have is radioactive antigen, it binds it again. And if you trap it and count it, it's always the same. But if you add plasma samples which have no radioactivity, now when it reassociates, it has a good chance of associating with an antigen, a hormone that is not labeled. So now that is a difference. And because you know the specific activity, you can quantify it. And that's how you can begin to now quantify blood samples, and that opens up clinical endocrinology. It now all becomes just not symptoms that you can see. Sometimes you can't see them, but the person is sick, obviously. But now you can go to the site that is affected and then develop therapies. So this is how it simply works. You can have many antigens, but if one antigen say this is the hormone, the antibody was made to recognize that hormone, then the antibody will bind it. You need to have a secondary antibody made in a different species that will recognize this type of antibody, because then you make a large complex that can be trapped under filter. This alone cannot be trapped under filter. And you can begin to appreciate that. So here you have your radioactive antigen, say radioactive hormone. You know the concentration you're using. You can quantify that from just the radioactivity emitted by the substance. You can count that in a scintillation counter. So if you only have radioactive antigen, radioactive hormone, you're going to saturate it. And when you trap it, you get 100% binding. That's what it means. So now you add unlabeled antigen. And this can be in vitro situation where you add some of the hormone. And what will it do? When the radioactive comes off the antibody, you can replace it with one that is not labeled. And in this case, you have enough unlabeled hormone to just retain 1/4 of the original 100% binding. So you get a decrease of 75%. You need a secondary antibody made in a different species. It could be generated in a goat, for example. And now you trap the complex. So now you are able to quantify how much hormone has been displaced. And you start with 100% binding. Then the more you add of the non-radioactive hormone, the less you trap on the filter, and thus you get this kind of curve until you have displaced nearly all the radioactive hormone. But you see, this is quantifiable, because what can you do with this? You generate this curve. This curve is called the standard curve. And what it does is it allows you to run an assay in a few experimental test tubes where you have different amounts of plasma. So if the person has an excessive amount of hormone, and that's what the problem is, then then you're able to read the displacement, the amount of displacement, which is going to be a lot, and then you just come here to the standard curve, you match it, and then you go down and you read from the x-axis the amount of hormone in plasma. You know the volume you took of plasma, so you can designate it in deciliters, for example. And now you have the ability to quantify a hormone. So if you're quantifying, for example, testosterone levels, then what happens is that if there are high testosterone levels, why is that so? Now you come in with another different red immunoassay and you quantify the levels of what? Luteinizing hormone, LH. That's what turns on the Leydig cell. And then you can also quantify the, the luteinizing hormone releasing hormone from the hypothalamus. So now you can say what you couldn't say before. Now you know which part of the axis is not working well. And then you can develop means to address the problem. So very powerful assay. I think this is slide 60. So we're going to stop here and then we'll go over. This is just an illustration of the displacement that goes on, which I made it as a cartoon so that you can kind of grasp. That concept is a very important concept here. So let me then go and explore questions.